Well, welcome everyone. I'm Joanne Geer. I'm the Executive Director for the Westchester Biotech Project. And welcome to our clinical research roundtable today on predictive analytics, which will address artificial intelligence, machine learning, and some other great tools. I want to introduce Michael Welling, who is our co-founder. Michael, would you like to say a few words?
I would. Thank you very much, Joanne. Good afternoon, good evening, good morning to everyone joining us from around the globe. Today's conversation is a truly inspiring one for me, primarily because it is such a great representation of our global community and global partnerships that we have been so fortunate to be able to forge, and you will see some of the fruits of that activity here today. For those of you who are joining us for the first time, welcome. The Westchester Biotech Project is a nonprofit 501(c) designed and focused on fostering collaboration around the globe with a primary interest in the researchers, data scientists, and engineers that really drive so much of the, I'm drawing a blank on the word, but that drives so much of the creativity and the solutions that we find when we talk about medical research, meeting the art of medicine, and ultimately best practices in patient care. As I said, partnerships are what fuel both our interest and our efforts. This slide here is a great example of a sample, if you will, of our partners that we have from around the globe. Some names that many of you are probably familiar with, other names perhaps you are not. And one of the things we encourage is not only to let us know if you'd like us to reach out and make an introduction to one of these companies, if there is an opportunity that you think might make sense for one of these companies, but also if you want to see your company name included on this list here. We welcome that, and truly it's these relationships and partnerships that drive and fuel all of the efforts here at the Westchester Biotech Project.
So we've been in business since 2017, so coming up on our 4th anniversary. And we have held many, more than 100 sessions of one kind or another, between in-person roundtables, virtual roundtables, symposia, and so on, and mainly set them up in order for us to listen to what we could do that would be of value to help researchers, data scientists, and engineers. And so the two that we are really focused on are Career Consortium, which is involved with all different kinds of training programs and so on. I'm not going to get into detail on those, but of which the Clinical Research Roundtable is part of this. I'm going to introduce you to one of the co-chairs in a moment. And then we are launching the Westchester Data Analytics Hub, which will ultimately bring together data scientists and engineers and researchers across discovery, preclinical, clinical, patient care, population health. We're starting with population health because we have the right guy, who's Paul Savage, who is an expert in business analytics, using medical records to look for emergence of health issues, disparities, and so on. So we've really got a great start there. But I want to take a moment. We share all this because if any of this interests you, we'd love to hear from you, and we'd be very happy to talk about getting you involved. I just want to briefly introduce Ellen Badenko. Would you like to say hello? So Ellen and Daria Krian are co-chairs of our clinical research cohort within our Career Consortium. And we're developing some really wonderful programs. Are you there, Ellen? I can't see you, but
yes, I'm here, Joanne. Thank you. Hello, everybody. My name is Ellen Badenko. I'm a registered nurse, a clinical research professional, currently with IQVIA as a clinical trials educator. Daria Krian and I are co-chairing Clinical Research Roundtable. We have been in clinical research on the forefront for many years, and Westchester Biotech is a unique organization because it brings into the fold literally everybody who has any connection to life sciences, any professionals. And with that, of course, the ultimate goal, like it was said earlier today, is to bring the good therapies to the patients, to improve the patient care. And our passion lies in raising awareness about clinical research, patient centricity, and bringing more professionals into the clinical research fold. And I'm looking forward to this program and other programs that Westchester Biotech presents. Thank you.
Well, thank you, Ellen. Your leadership is really making a difference in growing this community. And we will transition now for Kal to do his presentation. We have, I'll chat a bit while you're making that transition, Matt. Philippe Salphati, who you will hear from second, is also with IQVIA, and he got in touch with us when we first opened our doors about doing international programs, and is heading our Westchester Biotech Project Europe initiative. Just to share our process, we'll be listening to Kal, we'll be listening to Philippe, and then we'll start the roundtable. And we'll start off with Magali Topham from Montero Language Services, which is one of the key partners that we work with. And she will do what I'm going to ask everybody to do, is to introduce yourself, talk about your work, comment on anything that you heard in the presentation, ask any questions you like. We'll have a nice long time for a real roundtable, and I will literally go around the call and invite individuals to do that. Let's see, like Kal, looks like we are lined up there. Are you ready to go?
Absolutely. Thank you, Joanne. And thank you for this opportunity, and thanks to Westchester Biotech for the opportunity. Hello all. Today, we're going to talk about clinical research roundtable on predictive analytics and how artificial intelligence platforms are able to identify subpopulations. So to give a little bit of background, we all hear about artificial intelligence, machine learning, and we all get confused what it is. Is it robots walking on the streets? Or is it going to be just self-driving cars running on the roads? What is it? So artificial intelligence is actually more practical. It's not out of this world. It's more practical, and it's at the core of it. It is statistics and math. So you find out what makes sense in terms of math and in terms of statistics, what has got higher probability and things like that, and then you make decisions or take the next path based on those mathematics. So that's the artificial intelligence at its core. And to apply it, we have to find out different applications of it. And healthcare, with all the healthcare data that is available nowadays, is a prime place to apply artificial intelligence and machine learning, because it is not humanly possible to go through all the data points that are available on an even particular patient. If you go to the genomic level, it's thousands of unique data points. So unique, I mean, there are millions of data points, but unique in the sense every individual is identified by thousands of unique data points, and that is not possible humanly to analyze. So that's why we need artificial intelligence and machine learning in healthcare especially. So if we drill down into the clinical trial space, and this is the cost driver for the drugs that we see. We complain about the prices of drugs; it's too high. It's unfair and things like that. But if you think about it from the pharmaceutical company's point of view, they have spent billions of dollars. Let's assume that they had a molecule in the market after they have completed through all the phases of trials, and it cost them $1 billion, say, just a ballpark picture. But behind that, there are a lot of unsuccessful molecules. We don't hear about them, but those are the costs that the pharmaceutical companies have to incur, and they have to cover those costs for the unsuccessful molecules from the successful ones, because they have to run their company, they have to pay their employees and things like that. So if you look at the amount of trials, there are a number of trials that fail after phase 1. There are 35% of the trials that fail after phase 1. And this is fail fast, and this is quick fails. This looks at safety and a little bit of efficacy and stuff like that. And if we have more molecules that are going to eventually fail after phase 3, if they fail after phase 1, that's welcome, I would say, even though it's not desirable, but it's welcome than failing after phase 3. And then if you move along, then after phase 2, it goes up, and then over 65% of the molecules fail after phase 2. And after phase 3, there are over 35% of the molecules that fail. So if you combine all those, and for the people who do permutation combinations, they will find that it's the overall between phase 1 to phase 3, only 14% of the molecules succeed. So it's a fraction of a number, and that number is actually declining because all the low-hanging fruits are taken, and we are seeing that the drug approval and the success rate is declining. So that's a situation we are in. So how can we apply artificial intelligence in this situation? So let's assume you have data after, say, phase 1, after phase 2. So after each phase, you have data. If you can somehow analyze this data to find out, okay, these are the patient subgroups that are responding to the treatment, and these subgroups are not responding to the treatment, and then let's design the following trial based on that. So let's dig a little deeper into the data that is available. So here we look at, on left side is the clinical trial data that is available after each phase of the trial. And on the right side is the real world data. So how is real world data important in a clinical trial setting? So what real world data can provide is it provides the drugs that are already in the market, and it can provide information on a drug that has got a similar mechanism of action. So if you are working on a drug that has got a similar mechanism of action, and you have a drug that is already in the market that has got a similar mechanism of action, you can glean a lot of information from it. And then you can fine-tune your drug or fine-tune your molecule based on that information. And all this could be done using artificial intelligence, because the amount of data that is available on the real world side, the real world data is the data that is available from EMR data, electronic medical record, or electronic health record, the prescription and claims and billing data that is available, the product registry data. Those are the real world data, and you can glean a lot of information from it, and then apply those into a clinical trial setting, and then create your molecules with the information that is available in the real world, and then that will help you improve the probability of your trial success. And after each phase, you have some information based on phase 1 or phase 2, and then you can use those data to fine-tune your subsequent phases. To give an example, let's move on to the next slide. To give an example, that let's say there are 4 different treatments, and some treatments are working on some patients, and some treatments are not working on some patients. So if you can find out the patients for whom the treatment is working and the patients for whom the treatment is not working, what does it give you? It tells you that patients with certain genetic makeup, patients with certain comorbid conditions, patients with certain demographics, patients with certain who are taking certain concomitant medications, they are benefiting or not benefiting from a particular drug. So in a clinical trial setting, we will look at just one row of a treatment. We find out the green ones, that is the patients who are responding to the treatment, and we find out the orange ones for the patients who are not responding to the treatment. If you can find that, then you can only do a trial for the patients who are responding to your treatment and not include the patients who are not responding to your treatment. By doing so, the probability of trial success goes up exponentially. And imagine if you could do this in a drug, or in a class of drugs that are already in the market, and you can then segment and subsegment population based on this treatment A, B, C, and D. What happens is, especially in oncology, there are different lines of treatment. Patients start on treatment A, that is first line of therapy. If it works or doesn't work, go to second line of therapy, then third line of therapy. What happens if the patient who is going to benefit from fourth line of therapy is given that line of therapy right off the bat? Then this trial and error doesn't happen. So if an oncology patient who is seriously ill has to go through different lines of therapy, because that's the treatment recommendation, and they go through multiple lines of therapy, it didn't work, they change the medication, it didn't work, they change the medication again, and at the end they hit the fourth line therapy and it all of a sudden worked. So why can't we find out which patients are going to benefit from which treatment and give them that treatment right off the bat? And that's the essence of precision medicine. And all this can be enabled using this AI ML analytics, predictive analytics, and subgroup identification. So let's move on. So this is what we intend to create. You have different treatments on the column side. On the row side are the different adverse event and side effects. The green boxes, patients who don't have any side effects or adverse events, which is rare, especially in oncology, but then it's possible. But then there are other subgroups where patients benefit from the treatment, but they also may have certain adverse event or side effects. And if we can find out the risk benefit of the treatments, then we can delineate, okay, this patient should get that treatment because the risk benefit ratio is really in favor of the patient. So this is the essence of creating this grid. This is the essence of predictive analytics and artificial intelligence, and applying artificial intelligence in precision medicine and in healthcare. So I will pause there for a little bit and then see if you have any comments on this before we move on to the methodologies or underlying methodologies that we can use.
You know, Cal, what I'd like you to do is to continue on, because we're going to have a long discussion once we hear your presentation, but thank you.
Sure. So these are the methodologies that are playing underneath it. So on the top left, you will see there are multiple patients in a clinical trial setting. There are two groups, active group and the control group. In real world setting, there could be a control group if you're doing comparative effectiveness study of two different treatments. So what we intend to find is we intend to find the differential treatment effect. And this differential treatment effect is based on different patient attributes that are available. Some of the patient attributes I alluded to are genetic genomic biomarkers. It could be RNA expression data. It could be even demographic data, disease state of the patient, like stages of cancer, for example. It could be blood biomarkers like A1C levels, IL-6 levels. It could be even comorbid conditions the patients may have, or the concomitant medications they are taking. So there could be sometimes that patients, the treatment is interfering with the other medications the patient is taking, and they can't get off that medication. So those kinds of things we can identify. And then we can identify the patient subgroups on the right side with certain attributes. For example, let's say we are doing an oncology trial, and in this hypothetical oncology trial, some patients responded to the treatment, some patients did not respond, and some patients had adverse event. So as it turns out that this chemotherapy treatment that these patients were on, older patients actually benefited from the treatment. Patients who were 60 or older, patients who were 30 or younger had really adverse events, and the patients between 30 and 60, they had mixed results. So if we can, so this was the result of, say, let's say a trial or in a real world setting. So how do you make sense of it? So first of all, you have to find it. Who is, so if you look at the data in a combination, you will see that, okay, some patients are benefiting, some patients are having adverse events, and some patients are having mixed results. So what do we do with this drug? Should we allow it to continue to be marketed in the market, or we should stop this drug and take it off the market? But what would you tell to the patients who are benefiting from this treatment? The patients who are having adverse events, they will say that, okay, this drug is no good, let's take it off the market. So what do you do? So first of all, to find this out, whether this drug actually worked for somebody or if it has got any efficacy, you have to do this analysis to find out what are the patient attributes that are predictive of the effectiveness of the drug. So that's when we find out that patients over 60 benefited from the treatment. Then we go in and find out, okay, patients below 30 are having adverse events. It's focused on patient subgroups that are below 30. So now you have segmented and subsegment populations into different subgroups. And now you then go in and find out that this drug, and then find out how this drug might be working. You find out that this drug is really toxic. The chemotherapy treatment is really toxic. And the patients who had slower metabolism, the older patients, they had a smaller uptake of the drug in normal cells. As a result, they had less adverse events. However, the cancer tissues have higher metabolism rates. So the cancer tissues had a higher uptake of this drug, and the cancer tissues died off quickly. So that's how the patients over 60 benefited from this treatment. And the patients who are younger, they had higher metabolism in the normal tissues. So the normal tissues had a higher uptake of this toxic chemotherapy drug, and then they had adverse events. So now it started to make sense. So now you put a label on this drug, say that, okay, this drug is good for patients over 60, and maybe you can go a little bit deeper and measure the metabolism rate of each patient, and then give it based on that, and or administer it based on that, because it works when it works. So this is some of the examples, hypothetical examples that where AI/ML can be applied. So where you can go look at all different patient attributes that are available, and then see what are the attributes that are predictive, what are the attributes that are predictive of effectiveness or efficacy, what are the attributes for of adverse events or serious side effects, and then let's make precision medicine work. So that's on the top left. The bottom right is more of a in silico simulation. So you have trial data, and trial data is coming in, but trial data is expensive. If you have to recruit patients, you have to recruit sites where you do the clinical trials, and then after you do the trial, there is a lot of data that needs to be collected, collated, and it's expensive. That's why it costs over $1 billion to launch a product or drug into the market. How about we can use the data that is available, and then using that data while preserving the covariance structure of that data, create simulated data of simulated patients. So in other words, increase the sample size, and then see in this simulated world whether your new trial that you are planning to do is going to work or not. Because based on your prior trial, let's say phase 2 trial, you have some patient data. You use this patient data to increase the sample size to simulate a phase 3 trial. And you will have some patient inclusion exclusion criteria, because based on our prior conversation, you had patients who are responding to the treatment, patients who are older, patients who are younger, who are not responding to treatment or are having adverse events. You set the patient inclusion exclusion criteria accordingly. And then you simulate a new trial, and then you see now in this new simulated world whether your trial is working or not. And this simulation can also be done using AI/ML methodologies, technologies that is available today. It's not out 5 years out. It's available today. We just have to find applications of it. So these are some of the few applications, and these are the underlying methodologies where we can improve the probability of clinical trial success. There are other areas where this could be applied. We saw clinical trial as one on the left side, so which was trial design and simulation. If you go down a little bit, trial rescue. If you have a failed trial, we can help rescue it by finding patient subgroups who responded to the treatment better, and also identifying patients who had adverse events. So you can rescue the trial based on that. You can find out patients for early diagnosis, and I will go into it in a little bit more detail in terms of methodology, and Philippe can talk about its practical applications. On the top, safety surveillance. We are hearing about COVID. It's the talk of the town nowadays. Some people, recently there was a newswire article that said J&J vaccines are giving blood clots. To some people, it's not confirmed, but people are investigating it. How about we can find out which patient subgroups are having these blood clots? By doing so, what happens is you don't administer that vaccine to only those patient subgroups. Everybody else is fine. So we can do the safety surveillance using these artificial intelligence methodologies. On the right side, comparative effectiveness of different drugs for similar indications. We talked about that. Off-label use effectiveness. So if a drug is being used off-label, you don't promote it because you don't have a label for it, but it's being used off-label anyway. But passively, if the data is available, you can look at the data and then see whether off-label use is effective for certain patient subgroups. Because if there are patient subgroups who are benefiting from your drug, you probably want to get a label for it because then it can be used widely. So it's a benefit for both patients as well as for pharma companies. Down below, adherence tracking. If you have problems with patients not refilling their medication, which is not good for the patients and maybe not also good for the pharmaceutical companies, if you can identify which patient subgroups are going to not adhere to the treatment, and then invest your effort, marketing dollars to focus on those patient subgroups, then you can probably improve their adherence. So those kinds of patient attribute identification using AI/ML could be useful. And lastly, wearable devices. There is a lot of talk about wearable devices. How can wearable device predict a fall? How can wearable device predict some let's say ALS or any other ailment? So these predictive attributes can be used to identify different readings of wearable devices that can predict advent of, let's say, ALS, a patient who is going to fall, and then how it helps is you can use this learning to prevent fall. You can use this learning to intervene for patients who are progressing on a treatment, progressing on a debilitating disease, for example, ALS or dementia and things like that, and then you can slow it down. So it's adding life improvement capabilities or life improvement therapies early on, so that the patient can continue to be independent and productive for a longer duration of their life. And I would like Philippe to comment on this, because he has a lot of experience in commercializing drugs, and he has a lot of experience in applying AI/ML methodologies in commercial applications of drugs.
Well, thank you, Kal. And Philippe, I muted you earlier, so you need to unmute yourself. Philippe, would you introduce yourself, please, and tell us a little bit about what you do and share a little deeper information for us?
I'm Philippe Salphati. I am also a part of the IQVIA organization, which you have understood. Basically, this organization is entirely dedicated to life science, so to patients. I am in charge of globally addressing their performance, looking at patient Biotech Europe, in order to continue to expand this fantastic network of knowledge altogether. So if I may, then to come back to Kal point, yes, the launches of products, and when commercially we speak, the launches of product occurring 25 years ago and the launches of product occurring now have nothing in common, obviously, and artificial intelligence, predictive analytics, machine learning have been dramatically changed the way we operate and the way we target the patients. I just want to start with something that is currently happening. I know that in the US, J&J has made a pause in the immunization of his one-shot COVID-19 vaccine here because of some cases, rare cases of thrombosis. What if Kal and the predictive analytics could have been able in the real world to identify, or what if they are able in the real world to identify subpopulations which are more at risk of getting this type of adverse event with the vaccine? What if Kal and artificial intelligence and predictive analysis, we're able to identify the patient that should be using more BioNTech Pfizer vaccine rather than another one. This is a little bit a continuum now. It's bench to market. We leverage what has been done by Kal and his team to better address the needs of the patients. So I would say 4 things that I would like to discuss with you, and I would call that from bench to able. We are able to identify subpopulation here, subgroup, and subgroup with a lot of different characteristics, sometimes overlapping, sometimes not overlapping. The first one that, at least from a commercial, we say a commercial operation, but today it's a commercial operation mean also to, in a more sophisticated way, in a more personalized way. So four elements that I would like to discuss with you. The first one is what we call subgroup are basically collated into, or segmented, depending, into persona. This is the first thing that we are using basically when we are launching product and identifying patients on the market. The second is optimizing patient journey. The third one is education, awareness program, because if you have, for example, a new treatment on atopic dermatitis, which is basically a big transformation, most of the patients are not really eager to get the treatment. And you need to convince them specifically, not broad campaign, to go to these specific patients. Patient support program. So basically, when you have the patient, you need to continue to live with this patient, to customize your offering to this patient, to take care of this patient, to optimize the relationship of this patient with the treatment, to optimize basically additional use of these products. Kal was mentioning very briefly about off-label use. But when you are able with subpopulation to identify the way they behave, the way they work, how they comply on treatment, you are also eventually to identify new type of, I would say, sub-indication or other elements. So persona, patient journey, education program, patient support program, and new application are the 4 elements that are critical after launch to basically continue to improve the life of patients that are going to be under this treatment, or identifying the patient that should benefit from this treatment. And before we stop, I just would like to come to a more, I would say, practical element of operation. What we do today with artificial intelligence, machine learning, starting very early in the development, but leveraging all this information, is when we identify this subgroup, we are capable to have smarter discussion with HCPs. We noticed that, and I am going to take a very rare disease called arterial pulmonary hypertension, where the duration of life is today 7 years. When you are able to identify basically that the same practitioner providing the same drugs to 2 different patients will not exactly the same efficacy, the same compliance to the treatment. With that type of technology, I would say, we are able to train the HCP to better understand the patient, to even better speak to the patient in order to improve on the ground, on the field, the impact and the efficacy of each treatment. And we have seen that in this disease in a specific project that we did. That was really amazing. We can do that with the HCP, but we have done that also through patient support program with the patient, explaining to him what was the disease in a different way according to the different persona. This is basically all the services that artificial intelligence, machine learning, and / predictive analytics can bring to the life science and patients. So just to complete the very good landscape that was provided by Kal. So I will stop here for any question. Thank you.
Thank you, Philippe. Well, and we're going to move to our roundtable next. I'd like to invite Magali Topham to say a few words. So Magali is with Montero Language Services, which has just become a partner with us. We're going to talk about that a little bit later. Has continued to be a partner with us, but is growing our partnership. We're very excited about that. Magali, would you like to introduce yourself and make any comments or questions you'd like to do?
Thank you. First of all, thank you to our speakers. Very interesting. It's nice to also see it being applied to sort of current events that we're seeing. Just to give you a little overview of Montero, we're headquartered in Madrid, in Spain, but we do have now offices in New York, Westchester, more specifically. And we work with provide a range of language services, from translation and interpretation to localization and language consulting. We specialize specifically in biotech, life sciences, legal translation, and my specialty, which would be intellectual property. So this includes patents, trademarks, and all the surrounding documents. And we have about 40 specialized in-house translators, but we also collaborate with over 300 freelancers from around the globe with all sorts of different specialties as well. About myself, I'm the English translation team leader at Montero. I joined the company back at the beginning of 2014 and trained with them in IP and legal translation with the language combinations of Spanish and French into English. So that's a little bit of my background. Thank you.
Thank you. Did you have any questions or comments about the material we heard so far?
I don't for the minute. I'd love to see what everyone else has.
Thank you so much. Thank you, Cal.
Thank you, Joanne. Thank you
Philippe.
all.
Thank you.
Scott, Fritz.
Thank you.
Bye-bye, Joanne. Bye-bye, everybody.